Good afternoon, everyone. I think we better start now, but maybe there will be a few more students coming later. So, good morning, everyone, once again. Hope you're all having a wonderful day. This afternoon, we are honored to host a special talk featuring an eminent expert in microbiology, Emeritus Professor Dr. Ralf Conrad from Max Planck Institute for Terrestrial Microbiology, who will be sharing with you all his extensive knowledge on the topic methanogenic degradation and microbial metabolism of trace gases. So without taking up any more of your time, let's extend a warm welcome to Professor Conrad to the stage.
Yeah. Okay, thank you very much for the invitation and the kind introduction. I'm happy to give a talk about research which has been done over the last maybe 20 years and present particularly today methanogenic microbial communities. So communities, microbial communities, which in the end produce a trace gas methane, the atmosphere, which is important in the atmosphere. Now, it is about the atmosphere. Here, I like this picture, which I have taken from the internet, because it nicely shows the atmosphere. You see how fragile and small it is. It is just this little layer, this 10 kilometers from the Earth's surface. So the height of Mount Everest, maybe, which scratches on the top of the troposphere. And the troposphere contains 75% of all the mass in the atmosphere. The rest, the remaining 25% are a little bit higher now, maybe going out, slowly thinning out into space. But when we are dealing with atmospheric tracehouse gases, we talk about this shallow, thin layer around the Earth in which these gases accumulate and cause at the moment the climate change, which we more and more feel. Okay, now, what is that? It has shifted completely. I cannot show this slide, I'm afraid. It basically illustrated how much different trace gases contribute to the greenhouse effect. And it showed that the total greenhouse effect, what we experience on the Earth, is approximately 30 centigrades. So without any greenhouse gas greenhouse effect, it would be terribly cold on Earth. Now it would be below 0. And that of these 30 centigrades, most is natural, completely natural. And it is caused by water vapor and by CO2, mostly. And what we're talking about now, the dangerous increase, which we see at the moment, that's just at the moment 1.5 centigrades. And that is not, it's caused not by water vapor, because that is already saturated. It's caused by CO2, additional CO2, and other trace gases like methane, nitrous oxide, halogenated hydrocarbons, ozone, and others. But mostly, it's the most important one are CO2, methane, nitrous oxide. Here I show the methane, which depicts the cycling. We always have cycles, so we have a production of methane somewhere of different sources, which I show in a minute. There's approximately 600 million tons per year, 600 times 10 to the 12 gram, or 600 teragram, whatever you want to use as a unit, which are emitted into the atmosphere and cause average methane partial pressure of 1.85 parts per million. So it's very small, but it's effective. And this methane is removed from the atmosphere mainly through photochemical oxidation in the atmosphere itself. And a small part is taken up by soil and is utilized by soil bacteria, so-called methanotrophic bacteria. And this has been increasing over the last decades now, and is now approximately, it's more than 1.8 parts per million. And this increase is caused by an imbalance because the sources, the 600 million tons, are a little bit larger than the sinks. And as soon as this will be in a balance, then the concentration in the atmosphere will stop. And since the average lifetime of methane in the atmosphere is less than 10 years, it will be relatively fast process. So we will see it. It's different than CO2, which will take 100 years until it has stabilized. But methane, if we would do something about the methane cycle and stop it as a further increase, we would see a stabilization of the concentration in the atmosphere and therefore the greenhouse effect caused by the methane. Okay. These are the different sources. There are many papers now who have summarized this. So there's a summary which I used, compiled also from literature data, of course. And what I want to show you here is a number of different sources. And many of these sources are natural, so plants, wetlands, termites, ocean, gas hydrates in the oceans. It is natural. Rice fields, that's influenced by man. Ruminants, cattle, and so mostly influenced by man. Landfills, sewage treatment, biomass burning, fossil fuel utilized. That is all anthropogenic. That's one point. Another point which is important for me in the next, that is, what is microbial? So starting from wetlands, plants, it's not microbial, but from wetlands down to sewage treatment, that is all in principle the same processes. The degradation of organic matter yielding methane and CO2. It's all the same. There's the same process going on in termites, in ruminants, in rice fields, landfills, and so on. And this makes up all this part up to here. And only this 25%, so the remaining quarter, that is due to fossil fuel burning or to biomass burning, so to chemical processes, which can be relatively precisely determined because of the 14C content in the atmospheric methane. So this is the fossil fuel partner is old methane and therefore has a low 14C content, whereas the more recently produced, microbially produced or plant produced methane has a higher content. Okay. Now, I said it's the microbial processes are all the same now, whether they are in different environments. And in principle, that is the case. And that is a scheme of how the methane is produced from organic matter. So we have some steps which we can define, which are repeated in all the systems again and again. That is a hydrolysis step combined with a fermentation. So the first bacteria which tackle organic polymers in a rice field, it's mostly polysaccharides, for example, so rice straw and degradation products. They hydrolyze it to produce sugars, which they can use for fermentation to produce energy for life. And this fermentation yields fermentation products. Typically, these are various fatty acids, propionic acid, butyric acid, caproic acid, you name it, and also different alcohols, no? Mostly ethanol, no? But also isopropanol and things like that. No? And if the organic matter contains aromatic compounds, and we have benzoic acid and so on, something like that in addition. And besides these fatty acids and alcohols, there is always hydrogen and CO2 produced, sometimes format. Format is nothing else than hydrogen plus CO2. So, and the other main fermentation product, which is almost general, is acetate. And hydrogen, CO2, and acetate are the substrates then for methanogenic archaea. These are not bacteria, are archaea, but are microorganisms, little bugs. And say from methane, from hydrogen and CO2, or methane and CO2 from acetate. The acetate is split. The carboxyl group is transferred to CO2, is oxidized to CO2, and the methyl group is reduced to methane. It's a disproportionation reaction, which allows the methanogens who do that to gain their life, energy for life. Okay, these are the most important processes up to number 3 now. Now we have secondary fermenters, which also can use the fatty acids and alcohols and convert them again to hydrogen, CO2, and acetate. This is, we call them, like to call them syntrophic microorganisms or syntrophic bacteria. These are all bacteria, because they cannot do it by themselves now, because this process, this degradation process, is endergonic now. It consumes energy. Now. It does not produce energy, but only under standard conditions. So at 1 partial pressures of 1 atmosphere hydrogen, 1 atmosphere CO2, 1 molar of substrate, and so on. And the trick is now they have a syntrophy with the methanogenic archaea, which utilize the hydrogen. They remove the hydrogen, therefore the hydrogen partial pressure is very low, and then the reaction suddenly becomes now at a certain threshold level, becomes exergonic, and they can gain energy. No? And though it's actually a consortium of bacteria plus archaea, which is utilizing the ethanol or the propionic acid and so on. Therefore, this processes. So we only lack now 1 group of microorganisms that are the so-called homoacetogens. They have also a very peculiar metabolic pathway, the so-called Wood-Ljungdahl pathway, which is in some way comparative to the pathway in which methane is produced, but they produce acetate. So they can use hydrogen and CO2 and produce acetate, or that's something what methanogens cannot do, for reasons we don't completely understand. No? They can use sugars. They can convert the sugars in a fermentation reactions to acetate exclusively, only acetate as a product. Therefore, they are called also homoacetogenic bacteria. These are also bacteria. So these 5 groups now, and they do the job. Okay. Now I will show you several examples, different methanogenic environments, and explain you how this uniform process is modified in some way. There are little modifications, mostly with microorganisms, but also a little bit with the pathway. And I will go into detail. So rice fields is more or less that what I have shown in the scheme before. Exactly this process is going on in rice fields. And we can see this process after flooding of a rice field in different phases, actually. Can follow this in the phases. So we have phase 1 here, and we have the production of reducing sugars from the polymers, from the polysaccharides. So this is a very short phase, and then these sugars are utilized. You see they are going down, and we have the production of hydrogen, the production of acetate. I don't show the fatty acids and all this stuff. No methane production yet. This is phase 2. In this phase, also, this hydrogen is utilized by other microorganisms in the soil, microorganisms like denitrifiers, which reduce the nitrate. As long as it is available, of course, it becomes reduced in the end. Or they reduce ferric iron, as long as it's available. If it's completely reduced, then this process stops. Or sulfate. Some soils also contain sulfate. The sulfate is reduced to hydrogen sulfide. When then it's done, then sulfate is gone. And when sulfate is gone, then finally, and that is typical for phase 3, we start methane production from hydrogen. So hydrogen is utilized to allow methane production. Usually in this phase, acetate is not yet utilized, but it's still accumulating because the acetoclastic methanogens need a little bit longer to become activated. They are very slow-growing microorganisms. Needs a little bit longer. But then in phase 4, also the acetate is consumed, and then we have vigorous methane production. And you see hydrogen is then low. That does not mean it's not used anymore. It just shows that the production is leveled off by the consumption. So all the hydrogen which is produced is immediately consumed. And the same with the sugars. So the sugars are still produced by hydrolysis, but they are immediately consumed by producing hydrogen and acetate. And just acetate is not yet in steady state, it's still decreasing. But here in phase 5, everything is then in a quasi-steady state. It's not a true steady state, but it's close to a steady state. And so everything is low concentration, steady state concentration, and methane is produced at a constant rate. And at the same time also CO2 is produced. So at steady state, organic carbon is converted to CO2 and methane. So basically a disproportionation reaction. As mass carbon, which is oxidized to CO2, is also reduced to methane. Okay, this shows you the stoichiometry in more detail because it's quite interesting to do this. This is back of the envelope biochemistry, as we say, and everybody can do that. And you just have to make sure that carbon, hydrogen, oxygen, and all that is balanced on the left side of the equation as much as on the right side. Then you can do everything what you want. It's just a question whether it is realized in the end, but at least it's chemically possible. So here we have polysaccharides, which are fermented to acetate, CO2, and hydrogen. And if we have a polysaccharide, this is actually a glucose, so it's a unit of the polysaccharide, then we must produce 2 acetate, 2 CO2, and 4 hydrogen. No other possibility to balance the equation. Of course, we could produce propionate, and that we can also do that. It's just then extending the number of equations which we need to produce methane. So methane we produce from the acetate, that gives 2 methane and 2 CO2, and the hydrogen and the CO2 we are utilizing also to produce methane. So we have as a net effect, we have the polysaccharides again converted to CO2 and methane. Now we can learn something from these equations. We can learn, for example, that we have equal amounts of methane and CO2. That is almost trivial because I have already mentioned that. But what's more interesting is we produce 2 methane from acetate and 1 methane from CO2. So when this pathway is going on, we see immediately that the acetate is a more important substrate now, because more methane is produced from acetate than from CO2. And we can also make some assumptions about the CO2, but I have not the time to go into detail here, and therefore I just mention it. Now, when we look in rice fields again, now, and this is a relatively old study by a Chinese postdoc who used several soils from the Philippines, from the Erie stations, and analyzed the pathway of methane production via hydrogen or acetate by using a radioactive tracer. And with a radioactive tracer, we can determine here, he called it RH2. So that would be 30%. This line would indicate 30% of the methane being produced by hydrogen. And here it would be 20%, 10%, 40%, 50%. And this is plotted here in the rate of acetate turnover versus the rate of hydrogen turnover. And we see we are, with all these petty soil samples, in a range of 20 to 40%. So exactly that what we predicted theoretically from the stoichiometries. So we are happy. Another thing which is important in rice fields is where is the carbon coming from? Now, I mentioned now it's polysaccharides. Now, of course, one can ask this in a little bit more detail. Now, is this really true? It is true when rice straw is the main substrate. But in rice fields, of course, there's more than rice straw. In fact, we have three different sources of organic carbon. We have soil organic matter, we have straw, but we have also freshly synthesized organic carbon, which is excreted by the roots. Determining these percentages, which I show here, 60% actually from root now. So this is young carbon now. And it's also mostly sugars or carbohydrates, but it's also proteins, other stuff. No? So we don't know exactly, no? But it's different fatty acids and malic acid, citric acid, also acetate directly, but also sugars and different things. And we have straw, these are polysaccharides, and we have soil organic matter, this is old straw. So 90% of the straw is decomposed completely in the first year. No? So they are also studied by labeling straw and following that. 90%. But then what's left over, that is finally soil organic matter becoming older and older. No? And now I have not the time to explain how these studies were done, but they are relatively complicated. And therefore, I know only of three studies. One is from our group, one of the last one from Yen et al., and the others are from Japanese groups. And they are all relatively consistent in showing these percentages. No? So when plants are grown, then it's root exudation, which is most important. In the beginning of the season, when the soil is flooded, it's the old rice straw and soil organic matter now, which are contributing most. But late in the season, 60% by organic carbon excreted by the roots. Okay, another factor which is affecting the pathway of the methane production is temperature. So we studied temperature in quite some detail, and here it shows a rice field soil, so slurries now of rice fields, suspended rice soil incubated under anoxic conditions at 35 to 50 centigrades. 50 is relatively high, but we found that methane is produced up to 50 centigrades very easily, actually up to 60. No? and then nothing happens anymore. But rice fields also, they never experience under flooded conditions a temperature of 50 centigrades, only under dry conditions now have populations which can be active up to that temperature. But the populations, the community is changing at the temperature. So at normal in situ temperature, we have hydrogenotrophic methanobacteriales, methanomicrobiales, methanobacteriales, all hydrogenotrophic. We have the methanocellales, that is the blue ones, also hydrogenotrophic. And here the red and the green one, those are the acetoclastic ones. No? Methanosaetaceae, or nowadays they are again called methanotrichaceae. The taxonomists changed the names now. We can nothing do against that. So methanosaetaceae and methanosarcinaceae. And what we see here is that these acetoclastic methanogens decrease. The contribution is decreasing with increasing temperature. And so the big question was, of course, if there are no acetoclastic methanogens, but we produce from polysaccharide degradation acetate, not so much acetate. What happens with the acetate? There are two possibilities. Either the acetate accumulates, but it accumulated only a very short, brief time in the rice field soil. And then it was again consumed. Or what happens then? And the answer was that acetate is at high temperature consumed by syntrophic microorganisms, Thermoanaerobacteriaceae mostly. There's a family of bacteria which is able to utilize acetate anaerobically and produce hydrogen and CO2 in syntrophy with a methanogen, which is utilizing the hydrogen. That is very important. So these two reactions go together, and we have a conversion of acetate to methane, like an acetoclastic methanogen, but now two bacteria, one oxidizing the acetate and one using the hydrogen and reducing CO2. Okay, that is just the summary of rice field soils. So that plant material, root exudates, most under ecological conditions, mostly converted to acetate, which is the main substrate for methanogenesis, converted by methanosarcina and methanosaeta or methanotrix nowadays, and the hydrogen CO2 by methanocellales, methanobacteriales, and also methanosarcina can also use hydrogen CO2. They are also contributing hydrogenotrophically if they are present. Okay, now let's look at the different environments. A mire. Mire is a general name for all kinds of peat or other swamps. No? It is freshwater, a freshwater swamp. No? Yeah, I want to introduce another technique to determine the pathway of methanogenesis, which becomes available if you have mass spectrometers, which allow to determine the natural abundance of 13C and 12C in compounds. And the analysis of this ratio of 12C, 13C is usually expressed as a so-called delta 13C value. So that is just a number for the purpose which we need now. So we have the degradation of organic carbon, which has a certain C signature, a natural one. No? So nothing added. No? We can analyze this by taking organic carbon and measuring the certain C content. This is converted with a certain fractionation. A fractionation is going on because every kinetic reaction is preferring the light isotope. against the heavy isotope. And that's what we call fractionation. So as soon as they utilize the organic carbon, they will produce the acetate here, for example, which has a little bit lighter delta 13C. So it's a more negative value. And when this acetate is further converted to methane, so that is the methane produced from the methyl group of the acetate, it's still lighter. And the same happens with the CO2. And these are very characteristic values, these epsilon values, these are enrichment factors or fractionation factors. It is synonymous to that. And with knowledge of these values and measuring the 13C in the organic matter and in the methane and in the CO2, we can set up a mass balance. This is a mass balance. So the total methane produced would consist of methane produced from hydrogen CO2 and methane produced from acetate, and the fraction is given by this FCO2. So here FCO2 and the remainder is coming from acetate. So we can determine this fraction by knowing these values. The problem is we can measure that, but we cannot directly measure that. And the trick was we inhibit specifically the reaction here, and then we get this DMC, delta MC, directly by measurement because this is inhibited. And then we can find a value of the delta MA by two possibilities. One possibility is that all the acetate is immediately completely consumed, then there is no fractionation. And we can assume that it is 0. Or we have to assume that there is a fractionation going on. Then we have to measure the delta C in the methyl group of the acetate, which is a little bit time consuming. We have to extract the acetate and split it into the methyl and the carboxyl and determine the isotope. It's possible, but it's a little bit time consuming. Okay, when all this is done, so that is just an example from Finnish Fan Myers, when you want, so a mesotrophic and an oligotrophic. Here in the mesotrophic, that's the methane production rate, and they are decreased by adding different concentration of methyl fluoride. What is more important, that's the delta 13C in the CO2 and the delta 13C in the methane uninhibited. And with methyl fluoride, we only see those that methane which was produced from the CO2, and it is has a more negative delta 13C. Here in the oligotrophic, we don't see this effect. Therefore, here we have to assume it was entirely produced from hydrogen CO2. Because we don't see a strong inhibition of, not an effect on the isotopes with the acetate. So here we have a production of methane, which is approximately 40%, was approximately 40% hydrogenotrophic. So similar as in the rice field. So here it was exclusively hydrogenotrophic. So like at high temperature. So fans are something particular. So we can also determine the CO2 to methane ratio in what was produced. So we expect 1, no? Because it should be equal amounts produced from the degradation of polysaccharides. No? And in fact, here these are different Myers. In many, it is 1. But in many, it's more CO2 produced than methane. So more CO2, less methane. And these are here experiments. It's a control which is a little bit lower than 1. Here Textran added, or here it's iron 3 added So with iron 3, so CO2 increases tremendously, which is plausible because then iron reducing bacteria are utilizing the organic material and are completely oxidizing it, and no methane is produced. So anyway, what's happening in this fan is apparently that we have an external electron acceptor, and it is in these fans, it's not the iron, it is not nitrate, it's not sulfate, but it must be organic matter itself. And so the theory at the moment, and a lot of experiments shows this, it's humic acids. So we have some compounds in the humic acids which act as an electron acceptor. And so organic matter is oxidized to CO2. The electrons are transferred to this humic material and reduce it. And whenever these Mayas dry up a little bit, now, then the humic matter is again oxidized with oxygen. And so we have a cycling of oxidation and reduction of humic acids, and which allow the respiration, kind of a respiration process that organic matter is oxidized to CO2. So in Mayas, we have heterogeneous plant exudates, which are again converted to acetate and hydrogen CO2, but a lot is directly oxidized to CO2, and humic acids are reduced. And acetate is in these Mayas often accumulated. So we see over the season an accumulation of acetate, and at the end of the season, when the water level drops, this acetate is oxidized with oxygen, and is removed. Again, we have here methanosarcina and methanosar to utilize the acetate, but hydrogen CO2 is mostly utilized by Methanomicrobialis and Methanocellulase. I didn't show now a determination of the bacteria. Now let's go to lake sediments. Now I will show a little bit more bacteria here. In the lake sediments, again, we determined the fraction of methane produced from CO2 from this isotopes, natural isotopic abundance. So here plotted against the delta 13C in the methane. But what's important here, we see all these different lake sediments had a fraction of methane produced, which was higher than 50%. So hydrogenotrophic methanogenesis dominated. Again, the question, what happens with the acetate? So this was really point of concern to us here. And we studied therefore different lakes, and I had a collaboration with Brazilian colleagues. I got a lot of samples from them from the Amazon. And in the Amazon, we have basically three types of lakes or rivers. So the lakes are oxbow lakes, which are tributaries of the rivers where the water becomes stagnant. So we have clear water lakes. It's the south of the Amazon. They are mostly clear water lakes, crystal clear, wonderful. So it looks like that. So you can see the fish swimming. Then we have black water rivers. Now here, that is the Rio Negro, that is the Amazon. And the Rio Solimões, combining with the Rio Negro to the Amazon, that is at Manaus. This is tributary. You see it when you are on a ship. You see here the water of the Rio Solimões. It's white. It's really turbid. And of the Rio Negro, it's blackish clear, lots of humic acids. Very nice situation. So three types of sediments. They are shown here: clear water, white water, black water. These are methane production rates, logarithmic scale. So very high methane production rates in clear water, also in black water. In white water, interestingly, lower. Also, the methanogenic archaea, numbers of methanogenic archaea, also a logarithmic scale, lowest in white water, but clear water and black water high. Now here, this is a fraction of methane being produced from hydrogen, all over 40%. Okay. So what might be going on here? So that is our theoretical scheme. Complete degradation of organic matter. We have 33% of methane produced from CO2 and the remainder from acetate. What if the organic matter is not completely degraded? If it's already a little bit recalcitrant, it means only partially oxidized. Here a simple case where we don't change the oxidation state of the organic matter, but just degrade it incompletely. So we degrade it to some other compounds, to CO2 and hydrogen. And then we have methane production only from the hydrogen. And we can make a mixture of this and that, and then can create any percentage. And if we make it even more complicated, we change the oxidation state of the organic matter. So organic matter becoming more oxidized, which is left over, and then you produce more hydrogen. And that is nowadays possible. So we started now, and unfortunately, I don't have yet enough data of that. And I also had to stop research because of retirement. But now with high resolution mass spectrometers, you can analyze the organic matter in very much detail. And what you can do very nicely is you can determine CHO ratios, and this is the oxidation state of the organic matter. And what you see when you incubate sediments, you see a change in the oxidation state. So this actually happens now. And when in future, we may be able to trace it down to individual organic compounds. Now, what really happens with them, how they are becoming oxidized, because we can define formulas. No? So what we get, no? Classes of organic matter. That is all possible nowadays. Relatively big machines, big machineries. No? So millions of euro. No? So 10 Tesla magnets. So you come close now, you have to take off your glasses now and your mouthpieces and whatsoever, because the magnets are so strong. But we get a resolution of millidaltons, no? in the message. So amazing. Okay. So this is the explanation of this relatively high contribution of hydrogen, CO2. Now let's look at the microbial composition. So here, these are the archaea, and we see here this composition of the archaea in these different lakes. We see here Methanosarcina, this is these red ones, and Methanosaieta, these are these lila ones, no? So the violet ones. And so it's mostly the acetoclastic is Methanosaieta, and hydrogenotrophic are Methanomicrobialis, the yellow, and also some Methanobacterialis, but not quite so important. Among the bacteria, we see a lot of proteobacteria, these bluish ones, but also there is a Firmicutes here, this range. So these are Clostridia. So I cannot comment much on that at the moment, because these are just taxa. So we don't know what kind of metabolism they had. With the methanogens I can, but with the bacteria it's still a little bit difficult, because with the 16S RNA sequences, and these are pyrosequencing, you get the taxa, but this is not immediately an idea what kind of metabolism they might have. Now, lake sediments, we have organic material. It's partially decomposed, therefore a relatively high percentage of hydrogenotrophic methanogenesis, mostly Methanoscieta and Methanomicrobials. Now I will show you another wetland, little lakes, the canopy wetland. This was a recent discovery by one of my postdocs that came to me already with this discovery. He made it in another group, and then we continued working it in my group. That is, these canopy wetlands are tank bromeliads. Pineapple, typical for South America. So here it's introduced. Now we have also, of course, pineapples, but in Asia they are not native. So say South Americans, and they grow in the canopy and in the trees. They are vascular plants, highly abundant, epiphytic, and also down on the ground, not terrestrial. They collect water. They have these leaf rosettes in which water is collected, rainwater, and creates little lakes, and they assimilate nutrients from this water with trichomes. And they have in the water a diverse community of organisms, not only of microorganisms, also of little animals. And they emit methane. That's an interesting thing, because they are little lakes, of course, they also emit some methane. And so we have canopy bromeliads, we had mid-story bromeliads and terrestrial bromeliads. So we see here some pictures. I never have been on these expeditions. It was all done by Gunnar Martinsson, who was in Ecuador and in Costa Rica to sample. But you also can do this in the lab very nicely, because when you go to the Netherlands are commercially very famous for all the different flowers they sell. And they also sell all kinds of bromeliads now. So you can buy them there and keep them in the laboratory, in the greenhouse. So measuring methane emission rates and doing a statistical analysis, 10% of the methane emission is explained by the water content. As much water, more methane. The height above the ground is important, and the size of the plant is important, which explains together 21%, which is not so much. So there are more variability which is unexplained than which can be explained by these parameters. Nevertheless, methane production, terrestrial bromeliads, mid-story canopy bromeliads, you see in the terrestrial have the highest methane production rates. And this is a percentage was produced from hydrogen. And again, we see a high percentage produced from hydrogen. And then another thing I just want to mention is that when we incubate that and inject here certain CO2, so a labeled CO2, we see this label ending up in the methane. So some of the CO2 is assimilated by the plants, excreted into the water, and then stimulated methane production. So it's similar as in rice. So we have here the plant exudates or detritus producing acetate. Of course, acetate is produced, but it's barely converted to methane. It's mostly probably, that is our hypothesis, is mostly probably assimilated by the plants as a useful carbon source. And the remainder, the hydrogen CO2, is converted by mainly Methanomicrobials to methane. Now, a final environment I want to show you. These are drylands. So drylands don't emit methane. However, when we flood it in a wadi, there comes a flood, then also methane can be produced. So they are potential, like a paddy soil. No? When it is flooded, it produces methane. It's not flooded, it does not produce. No? So we have here in these desert soils, we have soil crusts, which are quite characteristic. And only where we find a lot of soil crusts, we have a heavy methane production in the end. So it's a very surface. It's only a few millimeters now here. So here is shown, it's 3 millimeters now, where we have chlorophyll A. We can measure an oxygen profile when it is moist, or yeah, that is what can easily be done. No? So these are cyanobacteria, many cyanobacteria, which are sitting here in the soil crust, but also lichens and algae. And here, when we analyze the microbial communities of this dry soil, we find a very poor community structure, not very diverse. Only Methanocella and Methanosarcina in most of these soils, not much diversity, which was quite amazing. And at this time, we were much interested in this Methanocella because this was a group which was discovered by us and by our Chinese colleagues and by some Japanese colleagues who taxonomically analyzed them in more details. And we were interested in this group and found that these were dominating hydrogen-consuming methanogens in these dry soils. So here we have desert soil, organic matter, again, acetate, and this acetate is mostly assimilated by the plants, and the hydrogen CO2 used by Methanocella. Another thing I want to show you here, we were interested in which kind of microorganisms were involved in the methane production. And here we used a technique, stable isotope probing, but not with carbon, but with labeled oxygen. We used oxygen, O18 labeled oxygen water, and every microorganism is taking up this water. So we actually label every microorganism, but only microorganisms which are active, of course. And in the soil crusts, now not all are active. When they are dry, they are inactive. So here, when the soil crust was wetted, 1 day, 1 week, 2 weeks, 3 weeks, and this is a control which was not wetted. We see that O18 was incorporated. What's here, that's a density gradient. You have to separate the nucleic acid in density gradient centrifugation. At the high density nucleic acid have incorporated O18. So we see in the dark and in the light an incorporation with time of O18, not in the control. And now we can use this DNA, this heavy DNA or heavy RNA, and characterize it by sequencing. And that was done here, and that shows now in the dark, we have over these weeks an increase of this green, and this green are Clostridia. So in the dark now, fermentative Clostridia are typically fermentative organisms, have used the organic matter and produced the material which was then subsequently converted to methane by the methanogens. So these are two different soil crusts, an arid one, a hyperarid one, both in the dark, both the same pattern. In the light, we see here this bluish one, which are cyanobacteria, so phototrophic organisms. Okay, I think that's it. Let's go to the conclusions. Polysaccharides are degraded to equal amounts of methane and CO2. In rice field soil, acetoclastic methanogenesis contributes the theoretically expected 2/3 with the methane. It's produced from straw, root exudates, soil organic matter. The microbial diversity is relatively large. We have all major families or orders of methanogens. But at elevated temperatures, we have an inhibition of the acetoclastic methanogens, which are replaced then by a syntrophic community. In peat soils, humic material apparently can serve as an oxidant. And in lake sediments, we have mainly hydrogenotrophic methanogenesis because organic matter is incompletely degraded. In rice field, as I mentioned now, more than 90% of rice straw is completely degraded in the first season. In lake sediment, it's already old. So when you go down in the depths now, it becomes increasing. I didn't show depth profiles, but that makes it even more evident now. And we go 10 centimeters deep in 100 years now. Sediment, that is quite common. In lake sediments, acetogenic, acetoclastic methanogens is mostly by Methanocyaeta and Methanomicrobialis and Methanocyaeta. They are the most important methanogens. Tank bromeliads are acting as little lakes. Biological soil crusts have also the potential to produce methane, but here we have a relatively low diversity community with Methanocellales and Methanosacyae. Okay, the last step I didn't show, mosses. No? Mosses also produce some methane. And here again, we have acetate accumulation and mostly methane from hydrogenotrophic methanogenesis. So these were co-workers associated with this study. Yeah. And yeah, that's it. I want to thank you. That's our institute. Yeah. Yes.
Thank you very much for Professor Ralf Conrad for a very give us for the lectures and share the experience, very good experience for us. Thank you very much.